Welcome back. Our next speaker will be delivering his talk online, Professor V.P. Upadhyay, who has joined us from Gorakhpur. Thank you, sir, for taking out your time to share your valuable knowledge with us. I would take the opportunity to quickly introduce Professor Upadhyay to the audience. Dr. Upadhyay, formerly the head of Department of Zoology. Sir, am I audible to you?
Yes, yes.
Okay. So Dr. Upadhyay, formerly the head of the Department of Zoology at DDU Gorakhpur University, is a distinguished figure in the field of sericulture. With a profound commitment to research and education, he has guided and mentored 18 students to successfully attain their PhD degrees. His scholarly contribution extend beyond the classroom as evidenced by his prolific publication record over 100 research papers in esteemed national and international journals. He has authored the well-regarded book Economic Zoology, which was published by Rastogi Publications. Additionally, his expertise in the realm of insect physiology is soon to be unveiled through his forthcoming publication, Insect Physiology, set to be released by Kalyani Publications. Beyond his research and academic pursuit, Dr. Upadhyay has left an indelible mark on Gorakhpur University's administration and extracurricular landscape. He notably served as examination controller and presided over the Athletic Association, showcasing in multi-faceted contributions to the university community. Sir, we'll be talking on the basics of sericulture today. Sir, over to you, please.
Yes. Okay. Let us start.
Yes.
Have nice day, everybody. First,
Hello.
I would like to define sericulture because my topic is basics of sericulture. I would like to define. People generally know that rearing of silkworm is sericulture, but it is not like this. Sericulture is the rearing, feeding of silkworm and the production of silk. It is an industrial word, sericulture. That complete is known as sericulture. This production of silk along with the rearing of silkworm. As I want to say something about the history of culture. About 3,500 years ago, this technique came into existence, and Chinese were knowing this technique about 2,000 years ago. But they were keeping this technique as sacred, as national sacred policy, because the gold was almost equal costly as silk. That's why the silk was flowing towards China from whole of the world. The silk, but the European tried to up and two monks were succeeded to take the silkworm seed from China, and they silk the silkworm rearing, or you can say the sericulture at the industrial level now existed in India, then Europe, then Australia, Russia, Japan, and Korea. You know that nowadays Korea is not Korea and South Korea, but it is completely distributed throughout. Now, projection please, number one. Projection. This is the cocoon of silk worm. Yellow is the cocoon of silkworm. It is also worm. Next slide, please.
इस पर क्लिक करके छोड़ देना, आपने ये स्पॉन्सर का आप देखो आगे पीछे हो जाएगा.
First one is silkworm. Second one is tussar silkworm. Then iris silkworm and muga silkworm. There are main 4 varieties of silkworm. Now, next slide, please. This is the Morus. First one is the Morus silva, second one is the Morus rubra, and third one is Morus nigra. You know that generally we rear the multi-voltine races, and that's why the mulberry, that is Morus that is mulberry, and that it feeds on the mulberry, and that's why it is known as the mulberry silkworm. That's why the Morus silva is most common, and Morus indica is also common for the feeding of silkworm, because the mulberry silkworm feeds only on mulberry leaves. That's why they are also known as monophagous insects, because they feed on mulberry only. Next slide, please. Life history of Bombyx morai. You can see that the males and females are there, but how can you distinguish the males and females? The males are with broader abdomen and females are with pointed abdomen. In this way, you can distinguish the male and female. Just after emergence, the male mates with the female, just after emergence. And after mating, just slide please, slide of life history. Yes, just after mating, the eggs are laid, and after egg laying, the female die. Just after laying, and one thing you must remember, that females are with very weak wings. They can't fly. Females can't fly, but males can fly. They have wings. That's why male reach to females and mate with them, and just after mating, female start egg laying. And these eggs, after 10 days, almost 10 days, 10 or 11 days, you can say 10 days of egg laying, they are hatched into first star larvae. The first star larvae, they are voracious feeder. They feed on the morus leaves because they are voracious feeder. Then they changes into second star, third star, fourth star, and lastly into fifth star. The size of fifth star larvae is almost 7.5 centimeter. They are quite large. And this fifth star larvae start spinning. Only they can form the silk. They start spinning, but just before spinning, they just stop feeding. Fifth star larvae stop feeding just before the spinning. And for spinning, there are spinning trays, especially designed spinning trays, and these trays, in these trays we keep the fifth star larvae, and the fifth star larvae start spinning, and thus they form the cocoon. These cocoons just after 10 or 11 days, as per climate and temperature, etc., humidity also, they start emergence from cocoon. Means cocoon they cover the pupa. You can say pupa. Pupa is covered by cocoon. Cocoons are the bed of pupa, you can say. And just after the formation of cocoon, after 10 days, they start emergence. And the male and females are emerged. In this way, they complete their life cycle in about 45 days. The larval period takes 20 to 25 days as per temperature and humidity. And in the lab, we can maintain the temperature and humidity, but farmers can't maintain the temperature and humidity completely. They try to maintain. There are certain methods they can maintain, but generally they can't maintain the temperature. That's why we rear the hardy species, the multi-boltine mulberry silkworm, that is Bombyx morai, and thus race is Nistri. It said Bombyx morai Nistri. There are 3 types of silkworm. First one is, next slide. First one is, this is the 5th star larvae. They are feeding on the mulberry leaves. They are 5th star larvae. But in this slide, we can see that larvae are more in number, but leaves are less. In this way, their growth will be affected. Larvae should be less in number and with complete mulberry leaves. In this slide, we can see that they are more in number, so their growth will be affected. Next slide. These are the cocoons and also the eggs. Here you can see the grainage management. The grainage management is very important process in the silkworm rearing, because for farmers we have to supply the silkworm either in the form of eggs or in the form of larvae. So the new rearers are always supplied with the eggs. The old rearers are given the 2nd or 1st star larvae, and that all may be done in the grainage. That's why for proper supply of seed means eggs. For the proper supply of seeds, the grainage management is very important. We can see that in the grainage management, in the slides, there are list figures regarding the grainage management, but we can see that the first part in the grainage management is the separation of pupae. We can separate the pupae from either from Nagahara machines, it is Japanese machine, or from hands. But we see that if the pupae are dead in particular batch, then that batch is separated and they may be discarded because they are not fit for further rearing or not fit for further production of silkworm eggs. First, we select the pupae and we can choose the pupae, male pupae and female pupae, and separate them. But it is possible only by the practice who are doing this job. We can't tell you how can you separate, but by practice you can separate the male and female pupae. And if you can't separate the male and female pupae in the grainage, the problem will be as just after emergence, male will start mating with the females. and if the male of the same lot will mate with the female of the same lot, then the seed will be not proper. It can't be given to the farmers. So they are first separated from each other. First male is separated and female is separated. So in the grainage management particularly, the males are separated after emergence and females are separated after emergence, and they are according to their batches. The male of one batch keep in the with the female of the other batch. The male and female, they mate, and after mating they start egg laying. Just they start egg laying. But it should keep in mind that male and female of the same batch should not be mate. Otherwise the egg will be not proper for giving to the rearers. So male and female of separate stocks mate, and just after mating, after 24 hours, about 24 hours, they start egg laying, and egg laying is for in the cellules. There is an apparatus, very small apparatus of server, cellules. They are kept in cellules and they lay egg inside the cellule. Inside the cellule, they can lay egg. Egg laying take place because the eggs may not be spread out. In cellules, they will be at certain place. They will not be spread out from each other. So cellules are used for egg laying, and just after egg laying, the eggs are kept in freezer at 4 degrees centigrade because they have to supply to the farmers, the old farmers. They are kept at 4 degrees centigrade. At 4 degrees centigrade, there will the development will not take place. So they will be as such at 4 degrees centigrade. There are freezer in the grainages, so they can be kept there in the trays, and after few days they can be supplied to the old farmers. But the new farmers should be given only first star and second star larvae. So in the tray where the eggs are kept in so many batches, they should be for proper emergence, for proper hatching, not emergence, for proper hatching of the eggs. The feathers, they should be touched from the feathers for proper hatching of the eggs. The proper hatching of the eggs takes place, and just after hatching, the first and second star larvae, they are very voracious feeder. So they feed and they can be controlled hatching. Their hatching should be controlled according to the availability of food. Food means the mulberry leaves. According to the availability of mulberry leaves, they are in batch wise, they are kept with the kept for hatching, and so just after hatching, they should get the mulberry leaves properly. So it can be managed very easily that when and how much eggs should hatch out. Just first and second star larvae always feed on very tender leaves. So very tender mulberry leaves should be chopped, or you can say should be cut into very small pieces. Then only first and second star larvae can feed. The third and fourth, and you can say the fifth star larvae can feed the hard leaves also. So they are directly given. The leaves are directly given without chopping. They may be given and they can feed on the hard leaves. So just after fifth star larvae, they they want to be secluded, and they reach to the secluded places for the spinning of cocoon. The spinning of cocoon takes place by the spinner. There is a hole spinner. They secrete through the spinneret, and they secrete silk through the spinneret. The silk is in the pasty form, but as they come out in the form of thread, it becomes dry. And thread, the elasticity of thread may be about 20%. The elasticity of thread may be about 20%. So the silk you can obtain for the industrial purposes. The further processing of cocoon may take place for other purposes. And for the silk industry, if you are managing the grainage, because we are here, grainage management, we are seeing that management operation. For management operation, something there is must for management operation. And these things are rearing trays. You can see rearing trays in this figure. I think there is no rearing tray. You can see the rearing trays and the machanas. Machanas means in which these rearing trays are kept at a place. The machanas and rearing trays, then the cellules, and then after cellules, you can keep the spinning trays, or spinning trays, or you can say chaki rear. This whole process is known as chaki rearing. So spinning trays, and in these spinning trays, you keep the fifth star larvae in these spinning trays and slightly slanting towards sun. For few time, you keep it towards sun. In this way, they can, about 3 days, the spinning is completed, and in this way, the formation of pupa takes place completely. In this way, we can complete the grainage operation and management of grainage operation. For the rearing at industrial scale, the grainage operation is very important. So one must know about the grainage operation. And generally, the grainage operations are performed by government institutes. In Gorakhpur, we have seen, I think that Kanchan must have seen, grainage in Gorakhpur. The maintenance of grainage is very costly thing. So in person, one can't manage the grainage. The grainages are always managed by government institutions. So grainage operation is very important. And I think that now I have completed my lecture. And what is this? Math emergency. Now that's described. Math emergency, okay? Grainage management, that done. Okay. And if anything, you can inquire from me. Very easy. Oh, okay. If anything, you can inquire.
Thank you, sir.
Okay.
For this insight.
If you want to inquire something,
Yes, sir. Your lecture.
not. It is very noisy.
Yes, sir. Okay. Okay.
Okay. Thank you very much. Thank you very much to all of you for it is very noisy. And I can't hear you. So I thanks you everybody for this patience hearing this unorganized lecture. You hear it very organized in very organized way. And I thank to the principal especially for organizing such type of workshop. And it is, I think it is of 1 week workshop. So, and lastly, I thank to Kanchan to connect me with this workshop. Okay, that's all. Thank you very much.
See my phone.
Okay. So may I Okay, thank you, and we regret for this all communication gap. I hope we sir has explained the process of sericulture in very simple words, and I'm sure that we have got we started getting a hang of the process. And now to continue with that, I'm delighted to introduce our next speaker, who is the second generation from Sir's team and happens to be a student of Upadhyay sir. So I'm also a very good friend of mine. Let me catch the line. Uh, we rarely get chance to introduce a friend. So Dr. Kanchan is working as an assistant professor.